Thank you very much, Ms. Bursett Harris. Friends, colleagues, ladies and gentlemen, before introducing the 2024 Szent-Györgyi Prize laureate, I would like to congratulate my friend, Dr. Marge Foti, for being awarded the AIM-High Prize. On behalf of the 2024 Szent-Györgyi Prize Selection Committee, co-chaired by Dr. Sujan Ba, it is my great honor and privilege to introduce Dr. Dennis Slamon, a superb scientist clinician and a leading figure in cancer therapy worldwide. The terms translational research, personalized or precision medicine, targeted therapy, or the expression of bench to bedside have all become household items within the biomedical community. The body of work of Dennis Slamon constitutes a common denominator for these terms. Dr. Slamon is a visionary pioneer in realizing that cancer is in fact an assembly of heterogeneous diseases. This notion was supported by the discoveries that breast cancer patients may differ from one another in the genomic, transcriptomic, and proteomic makeup of their cancer cells. The logical conclusion of this eye-opening realization was to develop therapies that would target specifically these variables. Dr. Slamon did just that. His discovery that the gene coding the human epidermal growth factor receptor 2, HER2, is amplified in up to 30% of human breast cancers and is associated with aggressive clinical behavior, was published in 1987. These transcriptomic results were supported by the finding that the amplification of the HER2 gene led to the overexpression of its gene product. Elegant experiments performed in 1989 by Slamon's group using genetically engineered HER2 overexpressing breast cancer cells provide preclinical evidence that a HER2 overexpression plays a key role in breast cancer pathogenesis. Based on these preclinical studies, Dr. Slamon set out to translate these discoveries into the cancer clinic. The question if the HER2 protein expressed on the membrane of breast cancer cells can be targeted by specific antibodies was positively answered. Anti-HER2 antibodies inhibited the growth of HER2 overexpressed in cancer cells in the test tube as well as in animal models. Importantly, Slamon's lab was the first to demonstrate that combining growth-restraining anti-HER2 antibodies with other standard and established therapeutic modalities, such as chemo or radiation therapy, was significantly more effective than either modality alone. One of the most famous anti-HER2 antibody is sold under the brand name Herceptin. This antibody drug was approved by the FDA in 1998. Patients treated with Herceptin alone experience objective responses with little or no toxicity. Importantly, patients receiving Herceptin in combination with chemotherapy had significantly improved response rates. Since its approval, nearly 3 million women with HER2 breast cancer worldwide were treated with Herceptin. The outcome is well known. There are numerous historical examples of active opposition to new ideas and concepts. This was also the case with Dr. Slamon's revolutionary discoveries of the 80s and 90s of last century. His novel approach to cancer pathobiology, namely that cancers with similar histology may employ different molecular mechanisms to survive and progress, deviated from the classical thinking of pathologists and therefore not easily swallowed. Equipped with unwavering determination and persistence, Dr. Slamon was victorious. I assume that in his acceptance speech, Dr. Slamon will allude to the recent and current endeavors to combat and cure breast cancer. Nevertheless, let me share with you some achievements of Dr. Slamon's group that the Szent-Györgyi Selection Committee deemed as highly important. The major subgroup of breast cancer, namely HER2 negative and estrogen receptor positive, is uniquely sensitive to inhibitors of cyclin-dependent kinases 4 and 6. The FDA approved in 2015 the use of a targeted inhibitor of these kinases that significantly improves the prognosis of high-risk breast cancer patients. In summary, Dr. Dennis Slamon discovered novel treatments that have saved countless lives and continues to play a major role in identifying and testing new anti-cancer agents for the betterment and welfare of patients. For these remarkable achievements, the selection committee of the 2024 Szent-Györgyi Prize selected Dr. Dennis Slamon to receive the 2024 prize. It is an honor to invite Dr. Sujan Ba, President and CEO of the National Foundation of Cancer Research and the co-chair of the Szent-Györgyi Prize Selection Committee, to join me on the podium.
Thank you very much. I am deeply honored to be selected as the 2024 recipient of the Szent-Györgyi Prize. If I were smart, I would just say thank you and leave the stage, because it can only be downhill. It can only be downhill from there. I think people have said some really wonderful things about the work we've done in our lab, but as Sid Arthur said, this work is not work that's done in a single laboratory. It's been done in multiple laboratories, not only in the US, but around the world. And colleagues around the world are every bit as much a part of this story as anyone in my lab or myself. So I want to acknowledge that right at the outset. We will talk about these two things briefly, and that is how these, we identified and validated these as novel targets, first HER2, and then the cyclin 4/6 kinases. Before going any further, I want to acknowledge someone. Acknowledgements usually come at the end. I want to do it right out of the gate. I graduated from an undergraduate school of about 600 students at Washington and Jefferson College. I majored in biology. I loved biology. I'm very glad I went to that school, but there wasn't much in terms of laboratory science done there. It wasn't until I matriculated at the University of Chicago that I was directed to a newly minted faculty member, Dr. Winston Anderson. And I want to acknowledge that my career was impacted in a dramatic way, and everything I learned about the scientific method and how to do science, not the theories, not what is read, not as what it taught us in class, but how to do science, I owe to Winston Anderson. It was Winston who taught me, never let your preconceived notions force you to fit the data to that notion. Similarly, when you do have data and you've been your own worst critic of that data, and it still tells you the same thing, believe it, no matter what the key opinion leaders tell you might be different. As I said, there are very few things that I can say were foundational to my professional career that are more impactful than what Winston taught me in the years that I spent with him at the University of Chicago. So Winston, thank you. He's here with us tonight. So if you've never looked at a breast cancer under the microscope before, you can see here 8 different breast cancers. They don't expect anybody to become a pathologist overnight, but you can see they're all except one is an invasive breast cancer. But you can see they don't look the same. They look different. And in fact, what our colleagues in pathology do, at one time I was giving a talk and I said our pathologic colleagues, and that was a big mistake. But what our colleagues in pathology do is do descriptive pathology that's been quite useful, decades-old information that tells us whether the cells are invasive or whether they're in situ, how the margins look, how the nuclear grade looks, et cetera. These are all important descriptive terms. And they told us that while we were calling this disease one disease, breast cancer, that really it might be heterogeneous. And before we break our arms patting ourselves on the back too much, we really should have recognized that from virtue of the fact of what was happening with our patients. As Siddhartha has already said, we were treating patients with sort of a one-size-fits-all approach of a surgery, radiation, and a combination of drugs, occasionally hormonal therapy, and the outcomes were dramatically different. And that should have been our clue that we weren't treating a single disease, but we were treating a spectrum of diseases. And that led us to beginning to think about how to look at that. And it was understanding when the molecular breakthroughs came through with biotechnology and molecular biology that we could begin to look at tumors not just under the microscope, but we can dissect them molecularly and try to understand, again, as Sid said, what is broken, what is converted a normal cell into a malignant cell. And it was with that in mind that we began to do our work that led to the identification of HER2. So the idea was that there is molecular diversity in human breast cancers that could easily and carry profound diagnostic and therapeutic information. So I'll take you just through this very briefly in terms of how we went about doing things in our lab. And again, as I said, there are multiple labs that are part of this story. But in our lab, what we had started actually in 1984, published our first paper in Science about the expression of this class of genes that were called oncogenes that were in fact, many of my colleagues that are prior Szent-Györgyi winners taught me about what these genes do. Tumor suppressor genes and the work that Webb did, Carlo Croce, and oncogenes, Peter Vogt, that these genes are important in the sense that they conduct normal growth activity. And our body's cells are growing every day, day in and day out. And it's only when something happens to those genes, either a direct mutation or an alteration in how they're expressed, that we can be off to the races with the process of abnormal growth that we know as cancer. So the way we decided to tackle this was to take tumors that had been removed for therapeutic purposes, and after the pathologists had made their diagnosis, in general, these were discarded. And we said with this new molecular technology, there's incredible information that we can pull out of those tumors. And so that's how we started the work we did. And we were fortunate enough to start a collaboration with Dr. Axel Ulrich, who at the time was at Genentech. And in Axel's lab was Lisa Cousins, who is with us here tonight. And Lisa gave a wonderful lecture earlier today. And She reminded she was the first to clone the HER2 gene in Oxel's lab. And then subsequently, Stu Aronson's lab identified a gene that looked like one of these oncogenes, as did Tadashi Yamamoto in Japan. And they were all working on this gene that had some interesting functions. It looked like a growth factor receptor. It sat like an antenna on the surface of the cell, received signals from the outside, transmitted them to the inside to tell the cell to grow. So we were working with Oxel looking, it's not like we knew HER2 was the problem. We had a whole series of genes that we had been looking at for a while, and we got 6 new ones from Oxel. And as I said, Lisa had recently cloned HER2 in his lab. And in an unbiased fashion, we went through our tumor banks looking for alterations. And we found this. Now, many of the new graduate students wouldn't even recognize what this is with the new technology. But for the scientists in the room, this is something called a southern blot. It's by today's technology, it's primitive. But each lane here represents a separate patient's tumor. And there are some, I think, 80 on this slide. And we're going in with a probe for the HER2 gene, as well as some of these other growth genes. And what we discovered was that despite we loading exactly the same amount of DNA in each lane, the intensity for the HER2 gene was dramatically different from patient to patient. And some had this very dark band that you can see at the top, and others had a very light band, barely visible. The light band that's very visible is what's normal. That's how it's supposed to look. It's this dark band that shows that there's been a mistake made. A mistake, again, as Sid alluded to, called gene amplification, where there have been multiple copies. So the Xerox machinery that's copying the genome gets stuck on a part of the genome in chromosome 17 and makes too many copies. Now, it's too many copies of a growth factor receptor. And the cell then goes on, does it make that growth factor receptor? So because the gene is amplified, it doesn't necessarily mean that it's expressed. So the next series of experiments we did was to correlate the amplification with expression at RNA and protein level. And that's simply what this shows, that every one of the ones that were amplified were overexpressing this receptor in the tumors. Now, this is not an inherited alteration. It is something that's acquired during the life of the individual. And we were very intrigued by having found this in about 20 to 25% of the breast cancers we looked at. But that led to the age-old and time-honored question of, so what? And the so what was the fact that we'd started with clinical material. So what we wanted to do was go back to the clinical data and ask, all these patients got best available standard therapy. Was the outcome different? And the outcome indeed was different. Those patients whose tumor contained this alteration unfortunately had a much shorter disease-free survival. They recurred more quickly. And equally unfortunately, many of them succumbed to their disease, usually within 3 to 5 years. So it was a very sobering thing. And that was the moment that we thought this might be an interesting thing to pursue further. The next question was, though, and this is the Kaplan-Meier curve. So many of you know exactly what these are, but for those of you who haven't seen it, when you start the observation period, 100% of the patients are alive. And as you follow them over time, you see that the survival changes, either recurrence or death. And there are 4 different lines here. All of these patients had breast cancer, early breast cancer. But there are 4 different lines: hormone receptor positive, another form underneath in the lighter blue is hormone receptor positive with other mutations that alter that pathway. In red are tumors that are so-called triple negative. They don't have the HER2 alteration and they don't express hormone receptors. And in the purple, showing the worst outcome, is the HER2 subtype in this particular study. So these patients indeed had the worst outcome. Now, when we published this work, initially there was a lot of controversy because other people were using different diagnostic antibodies to test for this protein, some of which were good, some of which were not so good. So there are a lot of reports saying this may not be true, but further technology evolved, allowed us to look even more carefully. And it was confirmed that this HER2 group did indeed exist. So there was a new subtype of breast cancer, not just hormone positive, hormone negative, but it was a HER2 subtype. And that led us to the idea that maybe we can do something about that. But in order to do something about that, we needed to go back into the laboratory. So as Isaac mentioned, we decided, and there are many different approaches people took. Some took cell lines that existed that had the alteration and others that did not. We decided to take cell lines that did not have the alteration and introduce it into those cell lines. So to artificially create in the laboratory what was happening in patients and ask, now they're totally matched, they're identical twins, if you will, except one has the alteration and one doesn't, and we're talking about a cell. Will they behave differently? And the answer was yes. We had a whole panel of cell lines that we could pick from. We picked the ones that were HER2 negative. There are some 51 breast cancer cell lines that we built in this panel. And we went ahead and engineered them to overexpress it. We also saw this in about 8 to 10% of ovarian cancers, and the same phenomenon was true. Those patients did even worse than ones who did not have the alteration. So it was a prognostic factor. So the question was, is this simply a useful flag for identifying aggressive tumors? Or is it tracking with aggressive tumors because it's playing some role in causing that aggressive behavior? And if the latter were true, that would mean something we needed to target or could potentially target. So what we did is we got a series of antibodies, and we took antibodies that had been produced at Genentech to study this protein. Brian Finley at Genentech made a whole series of 9 antibodies, I believe we got from then. We took antibodies from Amgen, another company called Triton Biosciences, and we made a few in our own laboratory. And actually, it was one of the ones from Amgen that had the most potent activity in terms of growth inhibitory activity. But before we got to that, if you look in the lower right-hand corner, when we were comparing the cells, in orange are the cells that did not have the HER2 alteration. In green are the cells that do have the HER2 alteration, and you're looking at the growth curve, number of cells in the Petri dish over a period of time. And you can see in green that the HER2 positive cells grow much more rapidly. There are many more, so it's more proliferative. We then took those cells, those same engineered cells, and put them into experimental animals, nude mice. And nude mice are called nude mice. Not many of the scientists know this is a very important tool for us because they're hairless. And they're hairless because in the mouse, the genes that code for hair are next to the genes that code for the immune function, for some of the immune function in those cells. Luckily, that's not the case in humans, given my hairline. So what we found was indeed when we implanted these cells, in the bottom is a littermate control that did not have the alteration, the same number of cells at the same time. At the top animal, we implanted the ones that did have the alteration. They grew much more rapid. They were more tumorigenic. And if you put it in a mammary fat pad and watched it there, waited for the tumor to form in the mammary fat pad, then remove it surgically, you would see that the metastatic potential was increased by 220% in the HER2 positives, which told us that this was something that really was tracking with the disease. So now we knew that it was associated with this bad outcome because it was potentially playing some role, and that meant we might be able to target it. Enter the antibodies. So antibodies have been tried before in a number of cancers, and they had failed. So the key opinion leaders said, this won't work. And not only key opinion leaders, but the grant reviewers said, this won't work. And a lot of the work that we did in this field was due to the collaboration we had with some scientists at Genentech, and it was a small group of scientists at Genentech. Lisa was part of that initial lab with OXO. Subsequently, Mike Shepard and his group was interested in TNF resistance and how that tracked with HER2 positivity. So there was a small group because the upper management at Genentech also believed that it wouldn't work at the time. So we went ahead and put this in these nude mice that were growing the tumors and found out when we put this antibody on board, one of the panel of 9, actually there were 3 that gave us inhibitory activity. The one that gave us the greatest activity was one called 4D5 that ultimately became the drug Trastuzumab or Herceptin. But this is from the mouse monoclonal studies where we showed if you had the antibody on board, you could completely suppress the growth of the tumor. In whiter, animals that did not get the antibody, and the tumor grew very rapidly. If you took a HER2 negative tumor, a breast cancer, it would grow just like you saw in the bottom animal. You could put as much of the antibody as you wanted on, it would have no impact. So it was specific to those that had the alteration. Hence, our concept that it was really going to be a targeted therapy. Armed with that data, we went to the FDA in collaboration with the clinical group at Genentech and asked for permission to do phase 1 clinical trials in patients whose tumors had this alteration, but for whom our current therapy had failed. It isn't the patient failed the therapy, we failed the patient. So our best available standard therapy had failed, they had a recurrent disease, and Sid also showed you Barbara Bradfield, who was in that first group of patients in the phase 1 studies. And it was, as we dose escalated, we had to start at doses less than we gave a mouse because this receptor is expressed in almost all normal epithelial tissues. So the regulators, the company, and we were worried, would we have some very bad toxic effects? And thankfully, as we dose escalated, we didn't see it. And it wasn't until we got to the highest doses that we began to see signs of activity. And that got everybody excited, even at Genentech, from some of the people who initially thought it wouldn't work, that maybe this could have legs. So we went ahead and did the clinical trial. This is graphically showing you, with numbers, showing you what Siddhartha showed you graphically, that we improved the response rate by 53%, the response duration by almost 60%, and the primary endpoint of the study was how long would you control disease, not have progression, and that went up by 65%. These data alone were sufficient to get the drug approved in 1998, and last year was the 25-year anniversary of the approval of Herceptin for metastatic breast cancer. But the concept was, could we do better if we used the drug even early? Again, as Sid mentioned, if we initially took this drug and gave it to patients whose tumors had this alteration and used it in a combination with their therapy up front. And remember the purple line. Patients had recurrence or death within 3 to 5 years. Now I'm going to show you the same kind of Kaplan-Meier data of a compilation of data from the literature that sort of looks at the history of breast cancer and how we've treated it. So you're now experts at reading Kaplan-Meier curves. 100% of the patients are alive at the beginning, and now if they're treated with surgery alone, and some may have received radiation afterwards, here are the outcome data. After about 13 to 15 years, 25% are alive without recurrence or having died. Pretty abysmal with that approach. It was Gianni Bonadonna, an Italian investigator, who realized that if we identified the high-risk patients, perhaps we could make this improve. And he used combination chemotherapy, a regimen called CMF, and indeed, this improved the outcomes significantly. This was not where we wanted to end. There are lots of different combinations that were put together, bigger, better, more. Sometimes that worked. Occasionally, we did worse. And there are variations on the theme of dose dense, et cetera, that made some small incremental improvements. But some of my colleagues would argue that you had to be sitting in the front row at ASCO to see the difference between those two. Meaning it may be clinically significant, it may be statistically significant, but is it clinically relevant? And then along came another Szent-Györgyi Prize award winner, Susan Van Herwitz. And it was Susan's work with taxanes that gave us the first, most effective chemotherapeutic drug for breast cancer, the taxanes. Not only used in breast cancer, but ovarian cancer. So that work made a huge impact in the outcome for the disease. And then there were variations on how to use the taxanes, of course, and we got some minor improvements with that. But when we added Herceptin to the regimen, remember the patients would have a recurrence within 3 to 5 years. This is what we saw. They went from having the worst outcome to having the best outcome. So the natural history of the disease essentially flipped. And this was very gratifying. Now the curve doesn't go out all the way because each of these drugs The CMF surgery was around the longest. CMF about the same amount of time. The taxanes less time in terms of the follow-up data on these. And Herceptin, the approval for early breast cancer happened in 2005. So we'll continue to follow these out. So that is what got us the HER2 story. And again, it was a collaboration of investigators all over the world who participated in the clinical trials, scientists and a small band of really believers at Genentech who kept us alive when upper management shut down the program that ultimately gave us the drug. But let me now finish up on the CDK4/6 story. How did that come about? So we were interested in looking at other targeted therapies, and we took the lessons learned from the HER2 story, and we ended up making a cancer cell panel, human cancer cell panel of about 650 to 700 cell lines. Why do we have 20 or more in each of these panels? Because we're trying as best the model can. No model's perfect, but we're trying to capture the clinical heterogeneity of the disease. If you're only looking at one or two cell lines, you may be only studying one subtype of a complex disease. So we have these panels. They've been characterized molecularly, full genome sequencing, proteomic expression. So we have all that data, their growth in 2D, in 3D, and their growth in animals. So all that's in a computational relational database. And then we can run drugs through it and see what kind of inhibition we get. And we ended up getting this drug from our colleagues at Pfizer. They were interested in running it through our panel because they had the drug. It was sitting on the shelf. It had been discovered multiple years before. It had been developed for lymphoma and myeloma, but they didn't see great activity. There was minimal clinical activity, but nothing like what they wanted, and it was suppressing some of the white cells, so they had it shelved. They said, we'll send this to you with a number of other drugs we want you to screen. So we went ahead and screened it, and we saw this. This is the target, by the way. This is the cell cycle, an incredibly beautifully regulated precision sort of activity when a cell divides, the things it has to do before one cell is allowed to divide into two, and the various checkpoints. Now we're not talking about immune checkpoints, but cell division checkpoints. And right at the beginning, this R point, called the restriction point, is retinoblastoma, which is sort of the first gatekeeper to make sure everything's okay and the cell can move forward and duplicate its DNA and then go on to the other phases. So there are other CDKs in other parts of the cycle, but CDK4/6 is right at this first checkpoint. Now, Winston's lesson to me about it's okay to have preconceived notions, but make sure you don't fit the data to the preconceived notion. My preconceived notion was, well, we want to look at this because we think maybe triple negative breast cancer, the red line that also was behaving badly, this might be an effective therapy for that. So we went ahead in a unbiased way, just ran the panel, so we were blinded to what the cell types were. It was all in the relational database. And we saw this. Each little dot is a separate cell in the cell line panel. And on the far left are all the cells, some 450 at this point before we got to 700 in the panel. And you can see this is a log scale. And the lower the cell is on it, the less drug it takes to inhibit their growth. So you want something that's really low on the chart. And there's a bunch that are stacked up at the top. They are completely resistant to the drug. And this is when we ran the CDK4/6 inhibitor in the panel. And we saw this tail in the blue, and then we were able to, because each cell has been identified in terms of which group it's in, we're able to walk from left to right. And sure enough, that tail was in the breast group. So as much as I was trying to diversify into looking at other cancers, we were back at breast cancer. So I said, well, great, maybe we've hit something for triple negative breast cancer. I could not have been more incorrect. This is the same inhibition data showing it in bar graph form. The bigger the bar, the more drug it takes to inhibit the growth. So on the far right, you see cells, you can put as much drug as you want on, it had no impact on their growth. In the middle are cells that had some moderate response, and in the far left are cells that were, even with small amounts, were completely inhibited in terms of their growth. When we unblinded the data, it was not triple negative or even HER2, it was the hormone receptor positive HER2 negative cells that were ones that were most responsive. So we then translated this into a clinical trial. We asked Pfizer for permission to run well, first we showed them the data, and again, lessons learned, and Winston's lessons he taught me was, we sent it to Pfizer and they said, well, that can't be right because we've already looked at breast cancer and the drug doesn't inhibit breast cancer cell lines. And they had a big panel too. And then the CCLA people at MIT said the same thing. They had looked at it. And I said, well, how did you do the assay? And they said, well, we do these high throughput ATPase readouts. So just the technical jargon is the cells grow for 2 days or 2.5 days and you get a read. Well, the problem is the hormone receptor positive cells that are HER2 negative grow slow, much slower. And they don't even double, for some of them, in a day or a day and a half. So they were calling cells resistant only because they hadn't gone through a division to show that they could be inhibited. And we were doing our assays 6 days long, not because we were smarter than anyone else, because we had made the same mistake earlier of looking at short terms and learned that you needed these long durations. So we told that to Pfizer. They repeated the experiments, called us back and said, we see the same thing you see. So I said, the only thing we want, it's not our drug, we don't have any IP on this. We want to be able to run the clinical trials. So they gave us the drug to run phase 1 trials to show it was safe, and then allowed us, once we showed safety, to move it into clinical testing. And this is what we saw. These are hormone receptor positive, HER2 negative cells. And in yellow are the ones treated with our best available standard hormonal therapy, and in blue are the ones who received this drug in combination. And this was a 20-month improvement in progression-free survival, which is the largest impact shown in that big group of breast cancers that have been seen since the introduction of hormonal therapy in 1975. These data alone were sufficient, phase 2 data, to have the FDA give conditional approval to the drug, as Isaac said, in 2015. And then just last month, it has been approved in early breast cancer. So where we moved is in blue what we could accomplish with the best available chemotherapy in this subgroup of breast cancer. We could get out to about 10 or 11 months progression-free survival. In yellow is what we can do with our best standard hormonal therapy. We can get out to 14 months. But when we add the CDK4/6 inhibitors, no chemotherapy, hormonal therapy and the CDK4/6 inhibitors, we're out to above 27 months progression-free survival. So again, that impact allowed for the approval of the drug, and it is now used globally. So what we've been able to accomplish, and when I say we, I mean we, the greater scientific community, the colleagues that I've had the good fortune to work with over the last several years, is we've made an impact on about 80% of breast cancers where the standard of care has now changed. HER2 patients get HER2 targeted therapy. It doesn't always work, but when it works, it's dramatic. And ER positive patients no longer get just hormonal therapy or hormonal chemotherapy. They get CDK4/6 inhibitors. And that's now being done globally. It's been approved by all regimens. So I'll finish by the acknowledgments. I want to acknowledge my colleagues at Genentech for the work we've done on the HER2 story, our colleagues at Pfizer and Novartis for the CDK4/6 programs. I've had the incredible good fortune to work with the National Breast Cancer Coalition and Fran Visco, which is an amazing organization that is not just doing advocacy. They want to be at the table looking at the data. They want to understand the rationale for the science, and they'll decide whether or not it's something they want to participate in. It's been a phenomenal organization, in addition to all the money they've raised for breast cancer research. My long-term colleague, Mike Press, who also trained with Winston Anderson from the University of Chicago, and then at Revlon, when we couldn't get grant money to do the initial work on Herceptin, and the very upper management at Genentech wasn't really enamored of this program, it was donor money we got from Ron Perlman, and it was Jim Conroy, who worked in Perlman's organization at the time, who went back to Ron and said, this is something you should invest in. And that was that first investment, when you talk about investors, of $2 million that allowed us to get through 3 years of the early preclinical work that proved that this was a doable thing. And then our network of trialists that I was able to form together with John Glaspie, who's been my partner at UCLA since I became division chief there. And then all participating patients. And finally, the group of 20 patients in the initial HER2 story and the 12 patients in the phase 1 CDK story who are not research subjects and are not numbers in a chart, but are colleagues in every sense of the word, as much as any of the people we work with in the lab or in the clinic. They're a part of what we do, and I thank them. And finally, I need to thank my family, and most importantly, without getting emotional, my wife. I would have thrown... Because I've become so obsessed with some of the stuff, I would have thrown my ass out in the street long ago, along with my bags. But she lived through all the highs and all the lows. So every scientist in the room knows when you see something in the lab that really excites you, you feel that high. It's a very personal feeling. When the lows come, your whole family gets to share the lows with you. So I thank my wife, Donna, for all the support. Thank you.